Okay, it's 1 p.m. now, so let's get started. Good afternoon, everybody. Thank you for joining us for Nour Aldossari's PhD defense seminar today. This is a public portion of her thesis defense. My name is Satoshi Ishii in the Department of Soil, Water, and Climate. I am one of the advisors for Nour, together with Brandy Toner. Because of the pandemic, our seminar is delivered via Zoom, but this does not necessarily a bad thing. It looks like Nour's family and friends are also joining from all over the world. If you wish, you may show your face, wave hands, and cheer up Nour. This is a time for you to do so if you wish. That's good to see.
I am Abir. I am a fellow student studying at the University of Oxford. Good luck, Nour. I wish you all the best.
Thank you.
Okay, good. From now on, please mute your audio. And there is a time for questions at the end of her seminar. You can also type your questions on our chat box so that she can answer these questions later. Okay, now let me introduce Nour. Nour received her undergraduate degree in biology from King Faisal University in Saudi Arabia and master's degree in microbial engineering from Ain Shams University in Egypt. Her master research is about thermostable alkaline protease produced by soil fungi. So she brought some fungal background here to the UOM. And Nour started working in my lab in spring 2017 as a research volunteer. Soon after that, I realized that she is smart and hardworking enough to be successful in the PhD program. She was officially admitted to the Land and Atmospheric Science program here at the University of Minnesota in fall 2017, and studied under the direction of Brandy Toner and me. Her research topic is about cold-adapted nitrate-reducing and cellulose-degrading fungi for nitrate removal applications. You will see parts of her research achievement from now. Nour, the floor is all yours.
Thanks, Satoshi, for the introduction, and welcome everybody for attending my thesis defense. So today I'm gonna tell you the story, which is about reducing nitrate from agricultural water using fungi. In 2008, the United States Environmental Protection Agency called for reassessment addressing eutrophication in the Mississippi River to reduce the size of the dead zone in the dead zone in the Gulf of Mexico. Eutrophication consists of nitrate and phosphorus. Today, I'm gonna talk about treating the surface water from nitrate. And here's showing the dead zone in Gulf of Mexico. And here's the picture snapped by NASA that's showing the algal blooms in the Gulf of Mexico. So this accumulation of nitrates, it's coming from the extensive network of surface ditches and subsurface tile drainage that have been built, and it's common practice in the upper Midwest in the USA. And that's due the drained soil type that we have here to allow great plant uptake of nutrients. Let's see how nitrate goes to the subsurface. When apply ammonium as a part of fertilization, ammonium gets oxidized to nitrates through a nitrification process. And there is excess of rain here in the upper Midwest, and there is a lot of snow, so it gets melted, and then nitrate gets leached and then goes to the surface water, and from there the problems start. So this practice, it usually happens during winter or late winter or early spring. So the temperature of water is between 5 and 15 Celsius. One approach to treat the agricultural water that goes through the subtle drainage is denitrification process. When there is some microorganisms, so they can breathe nitrate and then turn it to denitrogen gas. Let's see what's Woodchip bioreactor and its limitation. It's a subsurface trench filled with wood chips. And supposedly there is microorganism, but it faces some limitation. The first thing is the activity of microorganism gets reduced when the temperature drops down. We collaborated with Dr. Gary Friesen on improving the Woodchip bioreactor by doing two approaches, which is biostimulation by adding some carbon source or bioaugmentation, which is adding cold adapted denitrifying bacteria. It was like successfully removed nitrate at the beginning, but because when temperature dropped down to 4 or 5 Celsius, it slowed down the activity of denitrifying bacteria. Besides that, bacteria get washed out due to the water flow. Not only bacteria can perform denitrification, also archaea and fungi can perform denitrification. And actually, as you see here in this figure, fungi can apply or use nitrate in multiple methods. I'm going to explain each method that fungi can use. First method is denitrification. They use nitrate as a part of their respiration system. So they use nitrate in presence of electron donor like carbon. So they reduce nitrate to nitrite using Nour gene, which is nitrate reductase gene. And then from nitrite to nitric oxide using Nour-K nitrite reductase gene, and from nitric oxide to nitric oxide using the cytochrome B450 Nour. It's nitric oxide reductase gene. So this is the first process that fungi can perform. Second process is co-denitrification. They can drive nitrite or nitric oxide in the presence of ammonium to hybrid nitric oxide or denitrogen. And what type of ammonium that we have, we can know what type of end product we have. Is it denitrogen or nitric oxide? Third reaction they can perform is ammonium fermentation, which is similar to DNRA that bacteria can perform dissimilatory nitrate DNRA, dissimilatory nitrate reduction to ammonium, but it's different gene that they can use when they can reduce nitrite to nitrite to ammonium. Fourth reaction is chmo-denitrification. They can reduce nitrite or nitric oxide to nitric oxide or denitrogen in the presence of geobacter when they oxidize ferrous to ferric. So ferrous get combined with nitric oxide or nitric oxide to have nitric oxide and denitrogen at the end. So all this process, fungi can use it as part of their respiration system. They can use other process for other than their respiration system. And I'm gonna explain this in a bit, and it's important because some studies notice that this reaction and this gene and this reaction overexpressed during the denitrification process. Plant can reduce, or the plant root can reduce nitric oxide to make pathogenic fungi away from them. So nitric oxide is a toxin for fungi if it's in high concentration, but fungi can convert nitric oxide to multiple ways. The first way is to nitrate using FHB gene through nitric oxide detoxification. The second reaction, they convert nitric oxide to ammonium, and they use GS-Nour. Or they can convert nitric oxide to nitric oxide using the same gene, B415 Nour. So we know now that fungi can reduce nitrate, but in like multiple methods. So we can't call them denitrifying fungi. We can just call them nitrate-reducing fungi. And we use fungi to get added and improve the Woodchip reactor to remove nitrate at low temperature because these advantages. First thing, they have hyphae, so they can spread their hyphae in Woodchip reactor, so they get sick in the Woodchip and will help them to get retained. So they will not get washed out as bacteria have done in the past, and they will improve the nitrate reduction. Second thing, they are tolerant to cold temperature, so they will survive cold temperature as 5 Celsius that we need. Third thing, some fungi can secrete some enzyme extracellularly, so they can degrade that complex organic carbon that's in Woodchip, and they use it. They can use it as electron donor to perform denitrification process. So the overall of my thesis is to improve nitrate removal in Woodchip bioreactor by inoculating some fungi at low temperature. To meet this goal, we divided our chapters about that. So my second chapter is a review paper that review current knowledge that we have so far about fungal denitrification, and we can start from there. The third chapter is about isolating and characterizing a co-adapted nitrate-reducing fungi. And chapter number 4, we selected some fungi from chapter 3 to get inculated to Woodchip bioreactor mesocosm. And last chapter is I sequence a genome for selected fungus to know the gene that will participate in denitrification process. But now I'm gonna focus in chapter 3 and chapter 4. So chapter 2 is already published in soil biology and biochemistry. And the objective were, our objectives were, first one is to give you like overview about fungal denitrification and their end product. Also discuss their impact in the environment and summarize the recent method that we know so far to study fungal denitrification. And last objective is to give you some insight for future work. So please check it out if you are interested. My third chapter, the isolating and characterizing some cold adapted nitrate-reducing fungi at low temperature. Objective for first one, I isolated and characterized them. And then I confirmed that they were nitrate reducer by quantifying their end product using GCMS. And then I examined their cellulase enzymes. For chapter 3, I isolated my fungi from 2 different sources, and they were collected from Walmer, from here. And we got 349 isolate. To confirm that they were nitrate reducer, I transferred their culture into a slant, and then I made a cell suspension, and then transferred them to sealed file. Supplemented with labeled nitrate to quantify their end product using GCMS. Air phase gases were exchanged with helium And then they were incubated into different temperatures. And at the end, I quantified the antibiotic using traceability as I just explained. I got 0 colonies from Woodchip at 5 Celsius, but I got some from soil at 5 Celsius, and some more from soil and Woodchip at 15 Celsius. So the total nitrate reducer fungi were 91. I targeted their ITS to identify them, and I targeted their functional gene that were at their part for denitrification process. And it's known in fungi, it's Nour, fungal nitrite reductase gene, and cytochrome P450 nitric oxide reductase gene. And there were 35 Nour and 11 B450 Nour, and we got 8 that they have both genes. We feel like in our population, in the first place comes Fusarium. We got Fusarium in all strain and at all temperatures. Second comes Mortella. We got them just from soil in both temperatures. And third come is Mucor, and we found them in all sources and all temperatures. And all of them belong to Ascomycota and Mucormycota families. And we were test their cellulase enzymes. They were active. So we got 81% of them. In conclusion of that chapter, we also had 91 nitrate-reducing fungi, and they were able to reduce nitrate to denitrogen gas and was first report, and some of them were reduced nitrate to nitrous oxide, was hybrid and pure nitrous oxide. And they were able to degrade cellulose at what temperature. For chapter 4, I selected 5 strains from chapter 3. And then from these 5 strains, I selected just one of them based on the nitrate reduction rate and got inoculated to Woodchip bioreactor mesocosm. And we compared with a control without adding any strain. And then we examined the microbial activity or the population in Woodchip mesocosm. So to meet the first objective, I made cell suspension from each strain and get added to files that has glycerol peptone broth. I added label nitrate as electron acceptor, and the glycerol was electron donor here, and we exchanged gases with helium. Then we collected samples or gas samples every 6 hours for the first days, and then every 12 hours for the third day. As we collected gases samples, we collected liquid samples to measure ammonium nitrate and nitrite. And the strain that we selected was strain number 6, 7, 8, 10, and 15. And all of them belonged to Mortella hyline, except strain number 10 was belonged to Fusarium equisiti. Here, this figure, it shows that strain number 10 was our champion and was able to reduce nitrate by 1.4, or it's more than that, 1.5 milligram of N per liter per hour. And here it shows the liquid measurement of ammonium nitrate and nitrite for all samples. And I made 2 negative control vials. One of them was, I added label nitrate, and the other one nitrite. If I will start with ammonium and nitrite, as you see here, for all figures, we get it's about 0. Except the vial that was supplemented with nitrite, it's about the same all the time, indicating nothing happens in there because we didn't inoculate it with any strain. And if then we talk about, so there is no ammonium or nitrite in our samples. We talk about nitrate. I will start with the negative control. It stays the same. And then strain number 6, 7, 8, and 15 still, they reduce about the same, but strain number 10 was able to reduce nitrate by 50% in 42 hours. So they reduced nitrate, but in which form of gas? All of them were reduced nitrate to 30 denitrogen, which is denitrification. And we can see here in negative controls 0, so no chemo-denitrification isn't even happen here. And all strain is about 6%, except number 10 was able to reduce nitrate by 8% in 3 days to denitrogen gas. So we know now strain number 10 was the fastest in nitrate reduction, so was selected to the second and to continue the experiment. We prepare here 2 different treatment. So now we are mimicking the Woodchip reactor in the field. As you see here, the first inoculated, we made a cell suspension of strain number 10 and it was added. And we added a label nitrate as electron acceptor, and Woodchip was used as electron donor, and air phases was exchanged with helium. We collected gases every 6 hours for the first 2 days, starting from 0 time, and then every 12 hours for the 3rd day. Liquid samples was already collected at the same time point to major nitrite, nitrate, ammonium, and dissolved organic carbon. Woodchip was kept in every time point for qPCR purposes. So if we see here, the differences, I will start here with carbon. So we see here carbon was increased over time, and it's more in the inoculated than non-inoculated bioreactor mesocosm. So that means there is a sufficient carbon in the inoculated more than non-inoculated. That explains why nitrate was reduced more in the inoculated than non-inoculated Woodchip bioreactor. We got some nitrite concentration at the 0 time, but was reduced at 72 hours or after 72 hours. And we didn't detect any ammonium in our samples. So this figure, it summarizes how much nitrate concentration was reduced. As you see here in the control was 0.29 milligram of N per liter per hour. And in the inoculated, 0.43 milligram of N per liter per hour. So they reduce nitrate, but in which form? We get 4 forms here in this experiment. We got 25 denitrogen, indicating there is a co-denitrification because it's hybrid, as I explained in the figure in the beginning of this presentation. Now we got 30 nitrogen, which is pure denitrogen, and we got again hybrid nitrous oxide co-denitrification, and 46 nitrous oxide just denitrification. But both of them nitrous oxide was little amount of them, that means will not harm the environment. Here, denitrogen gas was produced more in the inoculated with our strain than non-inoculated Woodchip bioreactor mesocosm. And here is the same. We got more gases of 30N2 in the inoculated Woodchip mesocosm than non-inoculated Woodchip mesocosm bioreactors. So how, what's the total nitrate reduced at our Woodchip bioreactor mesocosm? Was about 85% in the inoculated and 62 in the control Woodchip mesocosm. And here's showing us if we do our experiment in the field, in which form our nitrate will be reduced. So as you see here, first come 30N2, and then it will come 29N2, and then 46 into all, and then 45 into all, which is about or close to 0. Okay. So I explained, like I kept the Woodchip at -80 for qPCR purposes. I extracted DNA and RNA. cDNA was synthesized from RNA to quantify the gene expressed during the denitrification process. We quantified the bacteria targeting their 16R RNA gene, and we got like it's kind of consistent all the time. I don't see any differences between the two treatment we used, but bacteria is more than definitely fungi. So the population of bacteria was more than the population of fungi in our Woodchip mesocosm. And if we see here, the population of fungi was increased over time in the inoculated Woodchip mesocosm more than non-inoculated Woodchip mesocosm. I designed some primer to target our selected fungi, strain number 10, Mortella hyaline, to see to quantify them after inoculating the Woodchip mesocosm bioreactor, and we got it's a little more in the inoculated than non-inoculated Woodchip mesocosm. That means maybe like this primer targeted same species, Mortella hyaline, because the differences between them is like slightly limit, like it's just slightly concentration. All right. As we target the population of bacteria and fungi and archaea, we target their functional genes that were responsible for the denitrification process here in fungi, and I will explain in a little bit in bacteria. So the first thing is NERK. So NERK gene is nitrite reductase gene that is responsible to convert nitrite to nitric oxide. And here is the population, and here transcript, that means that quantify the gene expressed during the incubation of Woodchip mesocosm. We can see here that population and in the inoculated was higher than non-inoculated over time, and the gene expressed or NERK gene expressed is higher in the inoculated than non-inoculated, indicating that it was enough carbon to motivate NERK gene. B450 gene. that are responsible to convert nitric oxide to nitrous oxide gas form, and they were increased over time in the in both treatment, but was higher in the inoculated than non-inoculated Woodchip mesocosm, and was expressed more in the inoculated than non-inoculated Woodchip mesocosm. Bioreactor indicating again there were sufficient carbon in the inoculated Woodchip mesocosm by our inoculated strain. So these genes were detected in bacteria. The first gene is Nour, which is responsible to convert nitrate to nitrite, which is Nour is referred to nitrate reductase gene. And I don't see here, it's kind of similar for both treatment. And here it might be the gene expressed more in the Woodchip mesocosm that were inoculated with our strain, indicating again there is sufficient carbon provided from the degrading of our from our strain that were added to the Woodchip mesocosm. Second gene that were detected was Nour, was responsible to convert nitric oxide to nitrous oxide. And as you can see here, no differences between both treatment, and even their expression is about the same. Last gene is Nour gene, and it's responsible to convert nitrous oxide to denitrogen. And they were consistent in both treatment and were expressed, is consistent, it's the same in both treatment as well. So in conclusion of this chapter, that we know now denitrifying bacteria and nitrate-reducing fungi can contribute to reduce nitrate in the Woodchip bioreactor mesocosm. And there is a sufficient carbon provided from fungi. And we indicated that from the inoculated Woodchip mesocosm, small amount of nitrous oxide produced. Adding fungi to Woodchip bioreactor can improve, overcome the limitation of Woodchip bioreactor at 5 Celsius. And our strain should be tested in the future in the field scale Woodchip bioreactor. Our overall conclusion, recommendation with cold adapted nitrate reducing and cellulose fungi is promising approach to enhance nitrate removal in Woodchip bioreactor at low temperature. And then I would like to thank my advisor, Dr. Satoshi Ishii, for sharing her knowledge with me and for her encouragement, Dr. Brandy Turner for her support and encouragement. Our my committee members, Dr. Kara Santley and Dr. Kara Rosen, for sharing their knowledge with me. Also, Saudi Arabian Cultural Mission in USA for supporting me. Dr. Gary Friesen for providing us a Woodchip and soil samples. Also, LAAS students, staff, professors, and other friends from different departments, all my friends and my family who attended today with us. Thank you all, and I'm happy to answer your questions.
Thank you, Nour, for presentation. Yeah, if you have any questions, you can unmute yourself or type on a chat box.
Satoshi, this is Satish Gupta. I got a question for Nour.
Okay, please go ahead.
Nour, you did a very good job. And as Brandy said, that's a lot of work. I'm impressed what you guys have done. Question is, most of your measurements were done 6 hours interval. Do you know what's the residence time of water in that bioreactor?
What's the resident time, is it?
Yeah, so is the water, let's say you take a cup of water, is that going to stay 6 hours or 72 hours in that bioreactor? In your case, you had it all the time in a given jar, but in the field, you have this bioreactor. The residence time has to be enough that you can make a dent in that thing.
Yeah, that's a good question. I don't know how they will react in the Woodchip reactor in the field, but I think if Woodchip will be closed, like control, so it's kind of control system. So I don't think the result will be changed to my result much.
Thank you. You did a good job. Thank you.
Thank you.
Hi, Noof. This is Melissa. Congratulations. It was a really interesting topic. I had a similar question to Satish, more about the kind of applied nature of this. If these organisms tend to thrive at 5 degrees C, would they potentially kind of die off every summer? And then would they have to be re-inoculated? How would that work?
That's a good question. I think the temperature of water goes up to 25 after that. So I think they will be tolerant to that temperature. So they will be survived, but I didn't testing how much nitrate they can reduce at 25 or 30 Celsius. But I assume they will do similar work, because I actually sometimes like I test my fungi, how they will survive at room temperature and can they thrive. So.
Thank you.
Yeah, thank you for your question.
So, hello, Nof. This is Gary. Great job.
Thank you.
Really, just a couple questions, maybe just to clarify some things. The chapter 4 experiment with the mesocosms, that was done at 5 degrees C?
Yes.
Is that right? That's, I was curious, and I might have missed it. There were bacteria in the wood chips, right?
Right, yeah.
Did you have any idea of how much of the denitrification was from the fungal activity versus the microbial?
Yeah, I did like actually an experiment. Maybe I can show you. Just a second. Yeah, so I did this experiment to know how much, fungi can contribute to nitrification in a wood chip. So I inhibited here fungal growth in the first vial. So I added here some antibiotic. And then I inhibited bacteria growth by adding streptomycin, and I inhibited both of them, and I didn't add anything. So we suppose all gases that will produce from here from both bacteria and fungi. And 50 ml was without adding any antibiotic and wood chip. And we found that fungi can contribute to the nitrification process by 40%. Yeah, at 5 Celsius.
Wow, that's great. I had one other question that you talked about the nitrous oxide being small. Did you quantify that? You know, maybe as either a percent, sometimes it's a percent of nitrate removed, for example.
Yeah, I think that what you can see here again, this figure is kind of, so this amount of gases produced to or nitrate reduced to nitrous oxide, we get like hybrid and pure, and both of them were close to zero. Just little amount.
You won't
This
give
answer,
you know, like 0.1%, 0.01%.
I think maybe 5, I will say 5%. Yeah, maybe less than 5%.
Okay, thank you.
Yeah, thank you.
No, you did a really great job. It was wonderful to hear your research. I'm just wondering, have you made any plans to patent your findings or to patent some of the strains that you discovered so that they could be used commercially in future endeavors?
Yeah, we apply for the patent, yeah.
Oh, that's great.
And
do you
thank you.
see any commercial opportunities for using this discovery about the denitrifying fungi?
We get contact from Denmark, but not yet contribution.
I think I have a question for Carl. I see he's on the line here. I think we should open the Wilmer bioreactors back up and try this strain out there.
I wish we could. It's a good comment, Gary. Yeah, I think that's the next step in this whole process. We have a nice laboratory experiment. We need to test it out in the field for sure.
That brings up a good question. How would you test that out in the field, Nour?
Well, I would the same, like do like cell suspension and like add some of my fungi there and test them in different season and see how they will react.
We've got a few bioreactor beds down in Faribault County. So what are you planning to do when your PhD is done?
That's a good question. That's huge. Well, I would love to test my fungi in this bioreactor then. Are they like Woodchip bioreactor or?
Yes, actually we've got 3 beds that they've been recently charged up, and so they have the same material in them. They're very large, however, but they're, you know, this would be same water source going into all 3. So there might be possibility to discern a difference.
Have you tried bacteria over there or what kind of source?
No, actually Satoshi had come down and gotten some of the original chips from that bioreactor for the isolation of the bacteria.
How is the nitrate removal then?
Well, it's a standard bed, you know, it's around 20% year round.
Let's give it a shot then. Try our fungi.
Okay. Maybe make sure the patent's in first.
Yeah, sure.
Okay, thank you for all the comments and questions. Are there any other questions, comments to Nour? Okay, if not, yeah, thank you, Nour, and great job for your presentation. Okay, so the public portion is done, and then Nour and the committee members will move to the different Zoom link for the closed portion. Maybe Nour, you can take maybe five minutes break or so to get some drink, and in the meantime, the committee members can discuss how to proceed.
Sounds good.
Okay.
Thank you.
See you soon.
Thank you all.
Yeah,
So thank you. Very nice. No.
Thank you, Gary. Thank you all.